Hello everyone, welcome back to our YouTube channel AgriAddict, a platform for all agricultural exams. This is our free lecture series for the IBPS A4 mains exam, and today our topic of discussion is plant disease management. Myself Ushasi Roy, I have completed my undergraduation from Bidhan Chandra Krishi Vishwavidyalaya, Mohanpur, West Bengal, and currently I am pursuing my master's in the Department of Plant Pathology at University of Agricultural Sciences, Dharwad. As you already know, if you have watched the first video that I have discussed about the first three principal methods of controlling plant diseases. Today in this video, I would like to discuss about the next three principles: protection, resistance, and therapy. So what do you mean by the term protection? It is the prevention of the infection by using chemical toxic barrier between the plant and the pathogen. That is the protection of the plant from the infection of the pathogen by use of any chemicals. There are various terms that are related to protection. First come fungicides. So the word side is derived from the word pseudo. It means to kill. So fungicides are the chemicals that directly or indirectly kill fungi. Fungistat. Fungistat doesn't kill the fungi, but it only inhibits or restricts the growth of the fungi. Antibiotics. Antibiotics are low molecular weight compounds. They are non-ribosomal origin and are produced by the actinomycetes and bacteria. This inhibits various subcellular processes of the bacteria. Example, penicillin and streptomycin. There are various other examples. We will discuss about it in the later slides. Antisporulants. From this word, you can understand that antisporulant. So, it inhibits the sporulation of the fungi, that is spore production. But it does not affect the hyphal growth. Bacteriocin. Bacteriocin are high molecular weight compounds. They are of ribosomal origin. They inhibit the growth of closely related bacterial strain. You shouldn't confuse between the two, okay? Antibiotics and bacteriocin. You can find the difference here. Since antibiotics are low molecular weight compounds, bacteriocins are high molecular weight compounds. It is of non-ribosomal origin and it is of ribosomal origin. Example of the bacteriocin is agrocin 84. It is produced by Agrobacterium radiobacter K84 strain, and this inhibits the growth of Agrobacterium tumefaciens. The other two chemicals that comes under protection is nematicide and insecticide. Nematicide kills nematodes and insecticides kill the insects. So for various diseases, nematodes and the insects act as the vector, okay, and they carry those pathogens and help in the spread of that pathogen. So nematicide and insecticide are also included in this protection. Fungicides can be classified on the basis of various parameters. Okay, so the first we will classify pathogen on the basis of the mode of action. It is of three types: protectant, therapeutant, and eradicant. What are protectant? Protectants are applied prior to the fungal infection. So, for a healthy plant, if we want to protect that plant from the pathogen, we will apply protectant, that is before the fungus has already infected the plant. Example is zinc. Therapeutant. So when to apply therapeutant? The fungus has already caused infection in the plant, but the symptoms are not yet visible. Okay, it is in the incubation period. Okay, so then therapeutants are applied for the controlling that fungus. Example, metalaxyl. And eradicant. When eradicant needs to be applied to a plant, when it has already formed the infection coat, when the symptoms of that particular disease are already visible from outside, then only we will apply eradicant so that it can cure the infection coat produced by the fungus. and it acts at the site of the application. Example, thydam. And on the basis of mobility within the plant, fungicides are classified into three types: systemic, contact, and translaminar. So systemic chemicals are those which has the mobility within the plant. That is, it can transport from one region of the plant to the another. So for example, this is the plant. We have applied the systemic fungicide on the leaves. Okay. So that chemical can travel from the leaf to the stem and then to the roots. Okay. So it will travel from the site of application to the other parts of the plant. Example are phenylamides. Phenylamides are generally systemic in nature. Contact. Contact fungicides are those that only act at the site of application. So the contact fungicide we are applying on the leaf, so it will act only on the leaf. Okay, only at the site of application. It will not travel from leaf to the other parts of the plant. And translaminar. So translaminar, the chemicals, like they only move within the plant organ. Like it is applied on the leaf, okay, in the upper surface of the leaf. So it from upper surface, it will travel to the lower surface of the plant. But they do not travel to any other plant organ. Example, phosphatyl aluminium. You can remember the examples, especially the translaminar. This is the uncommon one, and you need to remember this example. On the basis of use, fungicides are classified into various types. For example, seed treatment, like our propagating material, bulb, and tuber, seeds, all these things can be treated with thydam and captan. Okay, it is a protection of the planting material. Soil fungicides. We can apply the fungicides to the soil to reduce the inoculum potential that is present in the soil. So that can be pre-plant, that is before the planting of the crop, or post-plant, after the crop has been planted. So pre-planting chemicals, the examples are VAPAM and formaldehyde, and for post-planting, it's captan and thydam. Foliage protectants. Example is carbendazim, copper fungicides, fruit protectants, manip, tree wound dressings. Like in a tree, like the bark can be wounded, and that wound site can be the vulnerable site for the infection by a pathogen. Okay, so that wound can be dressed with Bordeaux paste and the localized application, for example, Litox. And apart from these three classification, fungicides can also be classified on the basis of the chemical composition. And the chemical composition classification is the most important one. So, according to the chemical composition, the first group of classification is copper fungicides. So, copper fungicides are classified in various types. The first comes is copper sulfate. Okay. The first name that we'll know about it is Bordeaux paste. It was discovered by Millardet in the year 1882. You need to remember the scientist name, and preferably sometimes the year comes, but like if you're comfortable with it remembering, you can do it. And the most important thing is you need to memorize the composition. It is 8 is to 15 is to 100. That is 8 parts of copper sulfate, 15 parts of lime, and 100 parts of water. So this is a composition of the Bordeaux paste. It was first used for the downy mildew of grapes. These are all the important information. There comes the Bordeaux paste. It is 1 kg of copper sulfate in 10 liter of water. Burgundy mixture. It was first discovered by Mason in the year 1887, and its composition is 4 is to 4 is to 50. That is copper sulfate with 4 parts of sodium carbonate and 50 parts of water. Stacient compound. It was discovered by Bewley in the year 1921. It consists of 2 parts of copper sulfate in 11 parts of ammonium carbonate. So don't confuse between all these compositions. You just need to memorize the name of the scientist who has discovered it. If you get the name of the disease for which it was first used and the composition. So these were all the types that comes under copper sulfate based fungicides. There are another type that is copper carbonate based fungicide. It is Schaubertia based. It was first discovered by Singh in 1942. It consists of 800 gram of red lead and 800 gram of copper carbonate in linseed oil or lanolin. Copper oxychloride based fungicides are Fungimer and Peronox, and cuprous oxide based fungicides are Blitox and Phytolan. So these are the examples of the various copper fungicides. And what is the mode of the action? The copper fungicides generally kill the spores of the fungi, and it is generally non-selective in nature. It is effective against the diseases like blight, downy mildew, and some rust diseases. And it is used as a foliar spray, wound dresser, and seed treatment. So you need to remember these names like Botox mixture, Botox pest, and the mode of the action and for which diseases it is used. The next comes the sulfur fungicides. It is ethylene bis dithiocarbamate and dialkyl dithiocarbamate group. The examples are Marneb, Zineb, Mancozeb, Ziram, Thydam, Nabam, and there are various other names. So, what is the mode of the action? It inhibits the action of various enzymes and it is used as a seed treatment and effective against diseases like blight, powdery mildew, and rot. So this is the most important. Like oftentimes it comes that sulfur fungicides are effective against which disease. So you always need to remember that it is powdery mildew. Okay. The other group is mercury-based fungicides. So now the mercury-based fungicides are not in use. Okay. These are completely banned due to the toxicity of the mercury. So there were other groups like mercury chloride, merfusan and mercil, mercurous chloride, cyclosan, and organomercurials, agalol, sericin, eritan, and emisan. So when it was rarely used for soil application, okay, and sometimes for seed treatment, but now it's prohibited. The quinone group has chloranil and dichlor. So what is the mode of action? It inhibits the action of various enzymes and it disrupts the electron transport system. It is used as a seed treatment. The heterocyclic nitrogenous compounds include cactan. Okay, it is also known as Kittelson's killer. Like it was first discovered by Kittelson, the scientist name. Folpet and Captafol. The benzene derivatives are PCNB. It is a short form of pentachloronitrobenzene. Brassicol, it is also called tadachlor, and chlorothalonil. And Dinocap. It is also, the commercial name is Carathen. It is used for powdery mildew. This is the most important because sometimes it comes like they will give you this name. The Carathen is applied for which disease? So powdery mildew. What is the mode of action? It inhibits the enzymatic processes, decarboxylation and citrate synthesis. The methyl benzimidazole carbamate (MBC) group. It has carbendazim, thiobendazole, and bavistine. It inhibits the DNA synthesis. It also disrupts the mitosis. And this group MBC is generally ineffective against the lower group of fungi. So lower group of fungi means oomycetes, oomycetes, right? So oomycetes generally cause downy mildew. So this group is generally ineffective, like it is not used against downy mildew. So always remember that MBC group fungicides are for higher group of fungi like Ascomycetes, Basidiomycetes, and Deuteromycetes. Okay, you can remember it by this. As we have already learned that if you have noticed, you have seen that copper fungicides are generally used for the lower groups of fungi. Okay, and this MBC group is for higher groups of fungi. There are various oxanthin derivatives, which is called carboxyl and oxycarboxyl. In the first video, I have already mentioned, but here also I would like to mention again that if you remember the abbreviations, that is CVS and OPR. So CVS, here it stands the chemical composition that is carboxyl, Vitavax V, and S. S is the disease for which it is effective against. that is mud and loose mud. And OPR, O oxycarboxy, plant wax is P, and it is effective against the disease rust. So what is the mode of action? It inhibits the respiration in fungi. The organophosphates group include ketazine and adipenophos. It generally inhibits the chitin biosynthesis pathway. The phenylamides include metalaxyl, and it inhibits the ribosomal RNA synthesis. The triazole group has tricyclazole, hexaconazole, and propiconazole, and they inhibit the melanyl biosynthesis pathway. Tricyclazole is often used for the protection against rice blast. Okay, you remember this. Okay, and the strobilurin group, it inhibits the process of respiration. So from this classification, you got the what are the types based on the chemical composition. You need to remember some of the names, and you also need to remember for which diseases it is effective against. Okay. So now we will learn about the various antibacterial antibiotics. So first groups comes is beta-lactam antibiotics. So now we will learn about the antibacterial antibiotics. So from this list, you will need to memorize the names of the various antibiotics, the source of this antibiotics, that is the name of the organism which produce this antibiotic, and the mode of the action. For example, penicillin, it inhibits the cell wall synthesis of the bacteria. From the polypeptides, the polymyxins and azoles, ketoconazole, they affect the cell membrane of the bacteria. The tetracycline erythromycin, this inhibits the protein synthesis. So from this list, you just memorize the important names of the antibiotics and their mode of action, and also the source from which it is produced. There are also various antifungal antibiotics. The name from them, aureofungin, is the most important one. It is produced by Streptoverticillium cinnamonium var terricola. It is effective against the fungi, and it disrupts the cell membrane containing sterol. Second is griseofulvin. It is produced by Penicillium griseofulvum. It is effective against the fungi, and it binds to the microtubule of the fungi. That is, it prevents the process of mitosis. So you just remember these names, the source, it is effective against which microorganism, and its mode of action. Like for plasticinius, it inhibits the protein synthesis. You just remember the shorter ones, like no need to have a detailed study. I gave the list, but you just sort it out and read it according to your convenience. The next principle that is used for managing a plant disease is resistance. So what is resistance? It is the genetic characteristics of the plant. It is the ability of the plant to withstand the attack by the pathogen. The variability in plants expressed by the response to external factors, including the resistance to the pathogen, is an essential requirement in any crop improvement program because it provides the sources of genes for the desirable characteristics. So here the desirable characteristics is resistance to any particular disease. So for that desirable character, the gene is isolated. The source of the gene is found, and from there the gene is isolated and introduced into the variety that is under cultivation. This variability is either naturally occurring or it is created during the crop improvement programs by the breeders. So, what are the sources of the genes for the disease resistance? It might be from a native or the indigenous variety or from any foreign commercial varieties. The older varieties that cultivation has been abandoned earlier, the discarded breeder stock, the wild plant relatives, especially from the areas of the origin of the plant species, and by induced mutation. The conventional breeding approaches which are used to develop resistant varieties are backcross method. So in backcross method, the popular variety that is under cultivation is used as the recipient, okay, and it is crossed with a with any variety, okay, that can be any wild relative or the older variety that has the source that has the source of the gene, that is the desirable gene, resistant gene. Okay, and this is used called donor. So we cross between them, and in the successive generations, probably in F6 or F7, we'll find the variety. Okay, this popular variety will obtain the gene from this variety, and also by selection of the resistant varieties. Development of the multi-line cultivars and use of various biotechnological approaches like tissue culture, viral coat protein synthesizing transgenic plant. So, resistance are of two types. It can be monogenic or vertical resistance, polygenic or horizontal resistance. So, what is vertical resistance? The plant is completely resistant to a few races of the pathogen. Okay, the plant will be completely resistant only to certain races of the pathogen, but this complete resistance is likely to break down easily if the new races of the pathogen attack the plant. It can be easily manipulated. Like this type of vertical resistance can be easily manipulated in the crop breeding programs. And horizontal resistance. The resistance evenly spread against all the races of the pathogen. The plant is never completely resistant or completely susceptible. This type of resistance is more stable than the vertical resistance because it has broader genetic base. Broader genetic base, why? Because it has all the resistant genes for all the races of the particular pathogen. And the last principal method for managing a plant disease is therapy. It is achieved by treating the plant with either chemical or heat or any other agent that will completely inactivate or kill the pathogen. This method is adopted after the onset of the disease. That is, after the disease has occurred to a plant, we will adopt therapy. So, therapy can be of various types like chemotherapy, use of the chemicals. So, use of the chemicals, we have already learned that we can use fungicide, okay, antibiotics. So, these are the example of chemotherapy. Thermotherapy, we can use heat. We have already discussed about it in our first video that there were various treatments like hot water treatment, hot air treatment, and radiation therapy. In radiation therapy, we can use UV ray or X-ray to inactivate the plant pathogens. Now we will learn about the integration of the plant disease management. It is also termed as IDM. So, what are the tools that comes under IDM? Biological control, that is use of the biocontrol agents, chemical control, that is the use of the various chemicals like fungicides, antibiotics, resistance, like we induce resistance in the host and use of the disease resistant cultivars, physical and mechanical methods, and cultural methods. Cultural methods includes the processes like tillage, flooding, and various other methods that we have already discussed about it. So, what are the features? It should be cost effective without any adverse effect on the environment and ecosystem. That is, it should be safe for the environment. Environment friendly methods. Procurement of clean and healthy planting material followed by proper seed treatment before planting. Clean field selection and proper soil treatment. Periodic logging of the diseased plants. Use of chemicals in an efficient way, that is need based application. That is the controlled application of NPK. Proper measures at the time of harvesting, proper storage condition for the harvested produce so that we can protect the harvested produce from post-harvest decay, and protection of non-target organisms. Because often we use the chemicals like fungicides and others, they disrupt the ecosystem, the soil ecosystem. Okay, they harm the other non-target or the beneficial organisms. So, protection of the non-target organisms should be the main feature. Okay, so these are the following features of the IDM. And the question arises that what is the need of the IDM? We have been learning about all the principles, right? Like only we can undertake biological control or chemical control. So why do we need to adopt IDM? That is why it should be in integrated approach. Too much reliance on the chemicals lead to resistance in the pathogen. The new pathotype develops and environmental pollution occurs. So, as we have said that the use of the certain chemicals, it leads to the resistance in the pathogen. So, we need to use other methods like use of the biocontrol agents. Okay, and we need to use like we need to adopt the cultivation of various disease-resistant varieties so that the resistance development in pathogen is reduced and the new pathotype development is also inhibited. And the reduced use of the chemicals will also reduce the pollution of the environment. To reduce the introduction of the new diseases to the crops. To avoid providing congenial condition to pathogen for disease development. Congenial means favorable environment conditions. So we need to modify the environment so that the pathogen cannot proliferate or multiply. To promote sustainable agriculture and to increase the effectiveness of the other inputs. So these are the features that comes under integrated plant disease management. I hope that today's lecture cleared your confusion and doubts and gave you a clear outview about the plant disease management and the principal methods that comes under plant disease management. So these are the two previous year questions that I have got from the question papers. So the first question is, which chemical is used for the seed treatment for the loose mark? Okay, so therum, carboxyn, Vitavax, alachlor, and none of this. So what would be the answer? It should be Vitavax because carboxyn is the name of the chemical group and Vitavax falls under carboxyn and I have already gave you that trick to remember that CVS, V for Vitavax and S means mud and loose mud. So the seed treatment which is used for the seed treatment, Vitavax. Which of the following biofertilizers do not provide nitrogen? Trichoderma viride, Rhizobium, Azotobacter, Acetobacter, and Aspergillum. So, what do you think could be the answer? Obviously, Trichoderma viride. I have already mentioned this name under the biocontrol agent. See, because Rhizobium, Azotobacter, Acetobacter, Aspergillum, all these four are biofertilizers, okay, and only this one is the biocontrol agent. And if you have any doubt about today's lecture, please feel free to comment in the comment section and we will reach out to you as soon as possible. And if you are willing to take preparation for any of the exam, you can enroll yourself in the online course that AgriAddict offers. It has Vijeta for the Target Batch 2025 for those who are taking preparation in IBPS A4 mains exam. Sarvata Batch for Target Batch 2025 for ICAR AICE PhD exam. Sarvashrestha for the Target Batch 2025 for ASRB NET exam. And for the PG aspirants, there are two batches. Shrestha for the Target Batch 2025 and Samartha for the Foundation Batch 2026. So enroll yourself in AgriAddict, learn from the best to be the best. And also follow us on Instagram, YouTube for further and latest updates. Thank you.